so I think we should start. Good afternoon, everybody, and welcome to Yuri Rodrigues' thesis defense. I have the privilege to chair the committee, which is composed of several people. Three of them are remote here. Dr. Suhita Nadkarni from India, she's there. Hello. Dr. Nicolas Brunel, Professor Nicolas Brunel, who's in Rome at the moment, I think, and Dr. Kin O'Donnell, who's also remote in UK. And the two co-supervisors, Dr. Hélène Marie, on my right-hand side, and Romain Welz, on my left-hand side, who are the two co-supervisors. So I propose, Yuri, that you explain to us your thesis entitled Unifying Experimental Heterogeneity in a Geometrical Synaptic Plasticity Model in about 45 minutes.
Okay.
And then you'll have a bunch of questions for you.
All right. Okay, thank you very much. Thank you very much for the introduction. So I will start. I'll not read again the title since it's already written.
Are you hearing him well? Sorry. Yes?
Yes?
Okay.
Yes.
Okay, so let's start by synapses. Synapses will be the center of this thesis. And synapses are the hub for neuron-to-neuron communication. When a presynaptic neuron spikes, this signal have to cross through all the synapses towards the postsynaptic neuron when you have to complete its journey, wherever it goes. And an interesting aspect of synapses is that they can regulate how the neural communication goes. And it is important part is that this function of regulating the neural communication is persistent. It means that after a long time, this fine-tuning of how neurons talk with each other will be maintained. And because of that, we'll be trying to understand these important structures that are considered the underlying basis for memory and learning. Okay, so knowing that these structures are important, experimentalists, they try to understand them because they have a great number of interactions. It is quite hard to understand them. And one part of the title that was trying to unify experimental heterogeneity, it reflects the problem that I'll be treating in this thesis. That is, it is highly likely that the methods used to probe synapses in one laboratory will be slightly different to another. And this will generate sometimes experiments that are not fully comparable. Because of that, we can have problems also using theory to understand this, how the synapses they learn. Because theory tries to make sense of data, and if you don't have data that's fully comparable, you may have problems. Okay? And in this thesis, I'll be trying to show how to derive a new synaptoplasticity rule to unify experimental heterogeneity. Okay. Initially, we'll be talking about synaptoplasticity. I'll be showing some fundamentals and the model that I built, because we're making a model to understand that, to explain that. And 2, we'll be deriving a new synaptoplasticity rule. In 3, we'll be exploring the geometry of the simulation to understand experimental heterogeneity. And 4, I'll finish with conclusion and perspectives. So let's start. Since we'll be doing a model to understand this problem, we want to have the most widely available set of experiments to work with. And one of those are the electrophysiologist experiments done in hippocampal slices. And most commonly, they measure plasticity outcomes by stimulating this region in the hippocampus called CA3 and recording the plasticity outcomes in CA1. So right here in the right, we see a representation that the presynaptic neurons are being stimulated, and the plasticity outcomes are being recorded in the postsynaptic neurons. However, we'll be just looking for a representation of a synapse. You have many synapses being connected in this pathway, but you're just looking to one that is thought to be representative of everything that's happening there. So here's our synapse. What what happens is when a spike arrives in the CA3 button, it will make vesicles that are filled with neurotransmitters. In the case here, we have glutamate. And this glutamate will be released in the synaptic cleft, and the glutamate will bind to AMPA receptors, okay, that we have in the other side, in the postsynaptic side. AMPA receptors, when they open, when they are bound to glutamate, they will let sodium to enter, and this will increase the membrane potential of the cell and will allow the signal to continue. So right now, we'll be looking to experiment that was proposed by Duendy and Lynch to study plasticity outcomes. And they show that when they do a stimulation in a given frequency, they will have excitatory postsynaptic potential amplitude, or just EPSP amplitude. They noticed that if they give a 100 Hz stimulation, if the signal is persistent, after a while, we can call this long-term potentiation, okay, LTP. And that means that you have a greater number of AMPA being incorporated in the postsynaptic side, and therefore, the signal will be easier to be conveyed from the presynaptic to the postsynaptic side. Otherwise, we can have a low-frequency stimulation, like for instance here 1 Hz, and this will decrease the EPSP amplitude and will lead to long-term depression, a persistent decrease in the number of AMPA receptor in the postsynaptic side. I don't know if it's okay. So right now, we just saw this experiment by Duendy, and they did using rats that range for, the age of the rats range for 40 days old to 100 days old. And in 93, around 15 years later, they discovered that depending on the age of the animal, the change in the EPSP amplitude will be different, will be more easier to induce LTD when the rat is very young. However, when the rat is almost adult, it's a little bit harder. And because of that, they saw very weak LTD. And a long time ago, it was a little bit discredited long-term depression. So here you see that depending on the age of the animal, you can have a different outcome. And this is part of the problem that you're trying to understand, how these different aspects, they will matter to predict what the plasticity outcome will be. So it is not only age that matters to modify plasticity, but also the external magnesium, the external calcium, and the temperature. And why this is important? Because when doing experiments in slices, you have to remove the part of the brain from the environment that is used to be, and you have to recreate this environment. So you have to put the intercellular medium there that's composed by many ions, for instance, magnesium and calcium. So, and you have to set the temperature that is sometimes not the same as the temperature of the brain. And this also can impact the plasticity outcomes. So therefore, this is important to understand how these parameters will affect plasticity outcomes. And now every time I show an experiment, I will put in this string that says the key parameters of the experiment I'll be showing. So in this case, you have the range of age, the value of the magnesium, the value of calcium, extracellular calcium, and the temperature. All right. Before, we are looking to direct stimulation. There was an electrode in the Schaffer collaterals in the hippocampus, and the Schaffer collateral are stimulating many axons. And sometimes you have or not a spike in the postsynaptic side. But when we discover the patch clamp technique, we manage to control precisely the time of the spikes. We don't let it by chance anymore. We can control the precise spiking times. And with that, we can inject a current in two connected neurons and manipulate when they will spike. For instance, when you inject a current in the postsynaptic neuron, you can produce a spike that's also action potential, and this spike will propagate it forward and will be propagated backwards. So this back-propagated action potential will be very important for us throughout this presentation. It's nice to remember. And now that you can control the precise time of the spikes, Bean-Poo in '98, they discovered that manipulating the precise time as you see here, between these two neurons, spike in time, they can change the amplitude of the response. So when they had a causal delay between the two neurons, they produce a LTP. And when they had anti-causal delay, like one pause, one pred 10, they had a long-term depression. But notice here that these little dots are quite distributed. It's almost, you can say that they're random. For instance, in the one pause, one pred 10 here, that's less than milliseconds between the two spikes, you have no plasticity, and also you can have a quite robust LTD. So there's some stochastic nature in how we induce plasticity. And that's been interesting to understand how this thing works, the nature of the plasticity. So why it is important to control the precise spiking times of the neuron is because also we can control the precise spike, the precise elevation in calcium. And with this precise control, we can induce plasticity. They discovered that in 83, without calcium, we cannot induce plasticity anymore, long-term depression. And they found that by inserting calcium chelator in the neuron and sequestrating calcium in a way that prevents calcium to trigger the cascade of events leading to plasticity. So let's remember that without calcium, you cannot have plasticity induction. Okay. Now that you know that it's stochastic, because I showed before in the BN pool example, we'd like to know what is the appropriate level of description to make a model of plasticity in the dendritic spines, in the synapses. If you see this small structure called dendritic spine, likely you will see that you have a very few number of receptors and ion channels. For instance, you can have in AMPA receptor that I showed before, you can have a small quantity of 0 to 100. So if you have one receptor failing, we may not have the full activity that you expect. So there's a lot of difference. If you have just 4 channels, one fail, it will be a great difference for the signal that you're transmitting. However, in the soma, it is nearly deterministic because you can have hundreds of receptors and ion channels. So we would like to know what is the appropriate level of description to make a nice model that, of plasticity. Because of that, we will be using a technique to model a framework called piecewise deterministic Markov process, or PDMP. So the PDMP, it allows to put together these two different dynamics, the stochastic ones and the continuous ones. So we have continuous variables, for instance, voltage or the calcium, and you have discrete variables, for instance, the AMPA receptors. So with that, we can define the PDMP as follows. We have a flow that will determine the evolution law of the continuous variables, depending on the continuous variables, the discrete, and the set of parameters mu. And also, to show the law of evolution of the discrete variables, we need a matrix Q to show how the jumps between the different states will have to occur, but also you need a transition rate depending on the continuous variables, the discrete, and a set of parameters. So right now, I'd like to put a small example here. That is a very simplified AMPA receptor that you have the closed states in C and the open states in O. So once you have glutamate, these AMPA receptors will be transiting from closed to open. And there's a very short example of action potential produced by AMPA when the glutamate comes, that this closed state will transit to open, and this will increase the membrane depolarization. And then you see more or less how the PDMP works. That is, you have the AMPA channel, the AMPA receptor opening, and then you have a continuous evolution of the membrane potential until the next jump occurs, that is jumping from open to close here, and then you have a change again of the membrane potential. Okay, now that you have the appropriate framework and the appropriate level of description, what would you like to do? So when I start my PhD, I came out with the first data set that I need to study to understand plasticity. It was provided by Tigare et al., 2016. And if you see here, you have an STDP protocol, spike time dependent plasticity, that says 1 pret to pos 10. And you can see also the voltage in the soma. And you can see here the calcium imaging the spine. And if you notice, during 1 second, this signal is very stochastic, okay? And after 20 seconds, you don't see any activity anymore, even if you have the spikes going on in the soma, and even if you have the presynaptic stimulation going on. So this is very intriguing. That means that the calcium is highly stochastic. It means that maybe you're going the right track to trying to have a stochastic model. And also it adapts both in the presynaptic way and the postsynaptic way. Okay? So right now I'd like to show bits of the model. You have 5 parts, like here. And first let's talk about the main source of variability in the calcium, because presynaptic release is a all or none behavior. You can or cannot have a release, and this will elevate or not the calcium in the postsynaptic side. So when you have a spike arriving in the presynaptic side, it will cause a vesicle to fuse and release glutamate. And this will make the docked vesicles, there's a pool of vesicles that are waiting for something to happen, to get a little bit less populated. And when this happens, the reserve pool will start to refill this pool of vesicles. And with that, we can have something called short-term depression. That means we are removing resources when you're stimulating. And then it becomes more difficult to stimulate. There's a little simulation here of the model using the same firing patterns from the last slide. That was 1 pre. And if you see here, within 20 seconds, as the previous slide, you had a strong short-term depression. It means that you are losing your resources, and you cannot stimulate further the postsynaptic side. And in black, you can see the number of failures that you have. That's a lot. Okay. So now that you have this great component that says that you may or may not have the calcium elevation in the postsynaptic side, we'd like to see another component of the model that's extremely important, and it will be composing the majority of the calcium signal that's so important for us to predict plasticity outcomes. Okay, I will not say about that. And this component is the NMDA receptor. The NMDA receptor, it carries in part of its current calcium, and is the major source of calcium. However, it can only open when it senses depolarization. So when the AMPA binds to glutamate, it will depolarize the spine a little bit, and this will let the NMDA to carry calcium in its current. And there's a small simulation here that shows the voltage driven by an AMPA receptor. And here you have the calcium signal in the postsynaptic side, and you see the elevation of calcium right now. But also you see they have other sources of calcium shaking this calcium variable. I will explain about that later. But first, I would like to focus a little bit more on the NMDA, because you want to incorporate experimental conditions, as I mentioned before. You want to know how you can code in the NMDA all these changes that can impact plasticity outcomes. So one of those are the magnesium. Okay, when the NMDA it senses a large depolarization, it can make its conductance is a little bit higher, and this will allow more calcium to enter together with its current. And this was shown by Yusti and Denk in '95 that they stimulate with an action potential, and they measure the fluorescence of calcium in the spine, and they saw that the EPSP was a little bit higher in terms of calcium. However, the sum of both together was smaller than the presentation of both at the same time. So you have a superlinear summation of calcium. It means that in MDA, when you have another spike in the postsynaptic side occurring, it can be stronger. It can allow more calcium to enter. And this depends on magnesium. And magnesium is something that you need to put in your extracellular medium to imitate the brain. What is the concentration of ions in the brain? However, we have other aspects, as I mentioned before, that we like to encode in the NMDA. That's the major source of calcium. One of those is the age. In NMDA, it will change its composition when you go from very young to very old. Here you see that when you are very young, you have a major composition of NMDA made of GLUN2B subunits. And these GLUN2B subunits make the NMDA very slow, a little bit slower. Sorry, not very slow. And once you start to get older, you have the majority of your NMDA composed by GLUN2A subunits. That's a little bit faster. Okay, so you have a change in the kinetics of NMDA when you are aging, when you are developing. Okay, so there's another aspect I'd like to include in to follow the experimental heterogeneity. That is the temperature. Since you need to set the temperature in the brain slice, it will affect the kinetics of the whole composition of the dendritic spines. For instance, in NMDAs, you have a much slower opening, also a slightly slower closing. And this will make this space of open slowly and close slowly will allow more calcium to enter. If you open very fast and close very fast, you have a shorter window for calcium to enter. So this also will impact the calcium that's so important for us to predict plasticity outcomes. And another and last part of modifications we are doing in the NMDA is the change in the conductance caused by extracellular calcium. If you have a very high calcium, the NMDA conductance will be reduced, and if you have a very low calcium, the NMDA conductance will be higher. Okay, it was long. There are some many other experimental conditions that will be included in other components of the model, but this is the main ones because NMDA is the major contributor to plasticity outcomes. So right now, I'll be talking about the second most important calcium source, but it's not only that. It is not just a list of things. This guy is that's called voltage-gated calcium channels. They answer to voltage changes, and they are able to incorporate the signal of the postsynaptic side. So the voltage-gated calcium channels have three types. They have many others, but I'll be including in this model only three types. You have the L type that answers for high voltage, the R type that answers for moderate levels of voltage, like around 40 millivolts, and T type that's extremely interesting because it answers for voltage that are close to the rest membrane potential. So even if you're doing anything, the T type is working, is shaking, so it flickers, as I like to put here. The T type flickers. Now I'll show a small simulation that represents the contribution of the VGCC to calcium. Now I'm showing a BEP, and here you see the calcium caused by VGCC, and you see how fast they open and close. And even after there is no voltage anymore, the T-type channel is open and closing. Okay, so it's really interesting because they are stochastic, and they will make a lot of difference in the resting conditions. So I spent a long time talking about calcium, but if you have a lot of calcium going on, it's also damaging for the cell. So in our brains, you have some safeguards of calcium that are, for instance, the SK channel and the GABA receptors. The SK channels, they sense calcium, and when they sense calcium and have a depolarization, they have a hyperpolarizing current to let potassium to enter, and this will shut down the synergy between VGCCs and the voltage, and also shut down the synergy between NMDA and the voltage. So it blocks calcium. When you have too much, it will activate. And also, GABA receptor that is permeable to chlorine, it will have a similar job. It will help to decrease the polarization of the cell. So here there's an example of showing what happened if you have a blocked GABA and SK channel, and if you don't have. And you see that without these two safeguards of calcium, you have a much higher, a slightly higher calcium entry. And so it means they are components that help to control the amount of calcium entering the cell. Okay, so it is really important to incorporate them because you don't want your calcium to grow without limits. Okay, now we manage to create a model of calcium that is sensitive somehow to experimental conditions, and you have inhibitory components. I'd like to know what to do with this appropriate level of description of calcium dynamics in the postsynapse side. So we come to look for the calcium hypothesis that dictates how synaptic plasticity is induced. The main two hypotheses for the calcium plasticity is the calcium amplitude hypothesis that says if you have high calcium after this threshold, you induce long-term potential. If you have an average amount of calcium, you have long-term depression. However, if you are under this threshold, you have no change. Another hypothesis for the calcium induction, the calcium plasticity induction, is the calcium duration hypothesis. It says that if you have a brief and high elevation of calcium, you induce long-term potentiation. And if you have a sustained calcium signal, you induce LTD. So the experiments that sustain these two hypotheses, one of the experiments are this. Okay, and meanwhile, I'll take some water just because my throat is dry. A moment. Much better. Okay. So there are many models that are based on these two hypotheses. One of those is provided by Graupner and Brunel, 2010. It says that you can predict plasticity outcomes just observing how calcium will cross two different thresholds. So here you have the SDP, that's 1 pos, 1 pred 20. And if you see, you have a part of the calcium elevations being slightly spending more time over the LTD threshold, but not over the LTP threshold. But for a causal pairing of this last example, you have most of it spending in above the LTP threshold. And using these different time spans over these different thresholds, they are able to predict a greater variability of curves, of synaptic plasticity curves. So they can generate virtually anything. However, it is hard to link to parameters of experimental conditions that we like to have. But recently, some experiments, some new papers have tried to connect this model, modifying it to adapt it to have extracellular calcium. So slightly, they're doing slowly some alterations of this model. However, they need to modify the thresholds to generate these many plasticity curves in some cases. Okay, because of that, we have also, we'll be trying later to look for other ways to predict plasticity that is not only calcium, even if you build a very detailed model of calcium. So because there's some certain limits, some experimental evidence is showing that you have some limits of it. So they show that if you have an average amount of calcium, you have LTD, and a high amount of calcium, you have LTP. However, as this work that you're trying to model in TIGRET 2016, they show that you can have the calcium fluorescence here, a very high amount of calcium for a plasticity protocol. However, you don't see any LTP here. See? So maybe use the calcium hypothesis is not the best solution for us right now. Because of that, we'll be trying to look to what comes after calcium. Okay, so one hypothesis that came out in 1989 by John Lisman is that calcium is controlled by interaction of two enzymes. One is KMK2, that's a kinase that's mostly related to long-term potentiation. Another one is calcineurin, that's mostly related to long-term depression and is a phosphatase. And it was shown that if you inhibit a calcineurin, you will block, you will induce only potentiation. However, if you inhibit KMK2, you will just induce long-term depression. Okay, and because of that, also we are trying to make a model to understand the calcium cascade, that what comes after calcium. And recently, the same John Lisman that made this hypothesis, he contributed to show that his initial idea that calcineurin affects the dephosphorylation of KMK2 is false. He found that there is no phosphatase affecting the decay of KMK2. And very important for us, because you're trying to make a model that's sensitive to experimental conditions, there's a new data that came in 2017 showing that depending on the temperature, you have a different activation of KMK2. So having this new data, we'd like to understand how it works. And we took an existing reaction system for KMK2, and we adapted to make it sensitive to experimental conditions. Okay, and here we have the simulation of KMK2 and calcineurin. And they have different decay times. This will be useful later for us to try to make a new interpretation. And right now, this is the summary of our model, okay? I know that it was maybe long and painful, but here's the summary. And we have the inhibitory components. We have the discrete release of the vesicles. We have calcium sources that are sensitive to age, temperature, magnesium, and calcium. And finally here, we have the calcium cascade, right? So with all this big model, we'd like to derive a new synaptoplasticity rule, okay? And now we are going back to that very first data set that I got from TIGRET 2016, and we try to use it, the information they have, to simulate the calcium cascade and see if we can discover something else. I'll just take a little bit of water, sorry. So, we simulated the protocol 1-PRE 2+50 that induces long-term potentiation, and also we simulated the protocol 2+1-PRE50 that induces no plasticity at all. And when we put them together, the enzymes that we simulated from our model, we see that they are highly overlapped. It is quite complicated to see what is the difference between these two. And this is matching data because the amount of KMK2 in the spine saturates very fast. So you see that it's quite similar here. So we like to try to, what you can do to separate these two curves. And we borrow an idea from dynamical system that is to combine these two enzymes in orbits. And we saw here that if you put them together, we are making an autonomous system without the time. We see that they have a little bit better separability. We see that after, this is a protocol that lasts 60 seconds, and we think here, in this point, they're a little bit more separated because you have more space, because you have a higher dimension. Okay, and since we have a model, we can simulate many times these curves, and you can notice that there is a preferable hotspot that LTP orbits they like to pass. And here's the time spent in this map, and you see that the LTP almost spent 1 second here in this place. And the no change, we can call it the undershoot, because it does not reach the LTP region, because we're assuming if they reach it, maybe there will be long-term potentiation. And then we can trace a separation between these two clouds, like this. Okay. So we are trying to design a Newtonian plasticity rule by mapping the position that the orbits like to spend in the calcinerine and KMK2 map. So let's put this separation here, and let's annotate that we put it here. Okay. So before this line is undershoot, and after is long-term potentiation. Okay. So far, I don't have anything. Let's have a look for another example. You have another example here. That is a SDP protocol that has a burst that's 1 pre 2 pos 10 and induces long-term potentiation. And another one is 1 pre 1 pos 10 that induces no change at all. And again, you have a great overlap, a little bit better in the calcinerine, but still a great overlap for KMK2. And then following the same trick, put them in a combined activity way, we see that you have a better separation, but right now it goes against the previous hypothesis that always put LTP above. And right now we have a no change protocol that's above. Okay? And if you see here, this protocol that's no change is over the LTP orbit. Because we have this model we can run many times and see where these curves like to spend most of this time in the map. And again, you have the cloud of the LTP spending almost 1 second here, and the cloud of the no change protocol spending most of this time here. And I will call it overshoot because it overshoots the region that we believe the LTP likes to spend most of this time. And maybe if this no change curve spends in the same place, it will be probably LTP as well. So let's put a separation, and let's annotate this separation. And right now, you can see that you have a little bit better, a better defined region for LTP, but we don't have yet for LTD. Let's have an example. Let's have a look for the only LTD case that you had in that data set. So, as you remember probably from the calcium duration hypothesis, the LTD case, you need to have a sustained amount of calcium for a long time. In this case, Tigare, 2016, they found that you have 2 pre 50 during at 3 hertz 900 times, it will have an LTD. So it's a much longer stimulation protocol when compared to 2 pre 10, 300 times at 5 hertz. That induces no change. So if you follow the same strategy, you see that, okay, maybe right now it's not so easy to separate them. And however, if you compare the time spent of these 2 different protocols, you see that the long-term depression likes to spend a long time inside this region. So right now, maybe it's not the position that's separating them, but the time spent. Okay, so let's make a separation that everything that's inside over this line is considered LTD if it spends a long time there. And if it spends a little time, it will be not considered anything. Okay, so we can draw this separation, and now we have a more or less well-defined region for LTD from here to here, and LTP. Okay, so the synaptic rule that I will show next comes from this experiment. So here again, we have the combined activity of KMK2 and calcineurin. It will cross the LTD region, and this will activate a threshold, okay? It is a region indicator, and this will go down, okay? So it means that I'm passing through this region. After I will reach the LTP region, and this threshold will go up. It means that I'm activating the threshold because I'm crossing this region. And again, after you stop to stimulate, it will just go back to rest because you need to cross the LTD region again. So we try to make the time that you spend in these regions to correlate with the plasticity outcomes. And for that, we need to find a way to say how much plasticity outcome you have. So we design a plasticity Markov chain that will start with 100 states in the no change, and depending the time spent in one region or another, you have a transition to LTP, or you have a transition to LTD. And this, in the end, will tell us the plasticity outcome. Right now, we have this. This is the structure of our SNAP plasticity rule. I'd like to see some examples how it works, because it took a long time to find these regions. And here, you have LTP example. And I will show how it works. You see that it's a very brief time that you need to spend inside the LTP region, and then you will have long-term potentiation. And the for LTD case that I was showing before, you need to spend a long time, like this. You can go out, you can go back. And then you have the plasticity Markov chain to predicting the plasticity outcome. So recalling that first example that I showed to make a counterpoint for the calcium amplitude hypothesis, we'll be trying to reproduce these plasticity protocols here, okay, using our new SNAP plasticity rule. And when you see, when you do many simulations, we can notice that you have a nice fit with the average. Also, we can reproduce the variability for this case. You see the data points in gray. But that's what I just said. Then, if you have a look for these two cases here, that are the causal and the anti-causal version of the STDP, that's one pre, two post, 50, and one post, two pre 50, you see that one will induce no plasticity, and another one will induce LTP. And Tigre was saying that probably Tigre is the person that did this data set, that they didn't manage to find the canonical STDP rule that for anti-causal spikes you have a long-term depression, and for causal spikes you have long-term potentiation. And if you measure the calcium fluorescence for these two protocols, you see that you have a very similar calcium elevation. So they are surprised by that, and they want to know why they cannot have the canonical STDP curve that I showed in the beginning with the bean pool example. And here's the simulation of these two cases. The one that induces LTP, as we know, it will cross through the LTP region. And the one that will induce nothing will be very fast throughout the LTD region. Because remember, you need to spend a long time inside this place to generate LTD. So right now, let's increase the number of pairing repetitions to see what happened. And here, if increasing from 300 to 1,200, you can have LTP. And for the other example, that's the anti-causal one, if you increase, you spend a long time inside the LTD region. That means that this is somehow related to the calcium duration hypothesis, that you need to spend a long time. You have to sustain a great amount of activity for a long time to induce LTD. If you do in a very short one, like 300 pulses, it will not be sufficient. And this prediction is offered by our model. Let's see other cases, because right now, we can not only control the pairing repetitions, but also the experimental conditions. And Tigre did the data with 300 pulses, as I showed before, but also we can now change the temperature. For instance, if we decrease the temperature, what will happen is from the no change, we will have LTP. And notice now that the kinetics of the enzymes are a little bit slower. And this little bit slower activity will modify, for instance, many requirements to induce LTP and LTD. If you increase the temperature, what will happen is that your receptors will open and close very fast, okay? And therefore, you have much less calcium entering. And you can see the geometry of these changes in the experimental conditions. For instance, here, I just increased 1.5 degrees, and you have a much less broader orbit, okay? And this one will not induce anything. And here's the prediction of the Tigoré experiment by different temperatures. All right, we spent a long time talking about SDP. Now, another important framework is the frequency-dependent plasticity. And this is the experiment that I showed in the beginning. This was the Deck and Bear. And they show that if you have a high frequency, that's 50 hertz, you will spend most of your resources to reach the LTP region, and after you go back slowly to the rest. And the other protocols, for instance, the 10 hertz, you don't spend a long time enough inside the LTD region. And the only one that does that, spend a long time inside the LTD region, is the 3 and 5 hertz. And we can see that simulating these curves many times, our model is able to match the variability of the data. And again, depending the number of repetitions, we can see how the LTD or LTP will be modified. And we can notice that before 300 pairing repetitions, you cannot induce LTD anymore because it's too short for LTD. And LTD, you need to spend a long time in that hotspot that I mentioned. Okay, notice right now that for 1 hertz, you have a very weak long-term depression because your rat is entering adulthood, it's 35 days old. And remember the first example that I showed of experimental heterogeneity, we are changing the age and we are looking how the LTD will be less or more strong. And here's an example of the no change with 35 days. And you see that it does not spend a long time inside that region. However, if you decrease the age, you have the kind of NMDA that's a little bit slower. So it opens a little bit longer time and allows more calcium to enter. And this little bit will allow it to reach the LTD region. And then you have LTD. So what happened is that the orbits for 5 days, it likes to spend most of this time here inside. If you increase the age, these orbits will have this location. And running many times, our model is able to match very nicely the data. And we can also see, not only for 1 hertz, we can see how different frequencies they will respond to age. And we notice here that you have a sink of LTD around between 1 and 3 hertz. And after adulthood, you start to lose this capacity of inducing LTD. But for LTP, is slightly different because you don't have, it's not easy to induce LTP for using this protocol from the Deck and Beer 93. In 2002, Schuval, it suggested that the NMDA decay will modify the sliding threshold. It means that the precise frequency that separates the LTD case to the LTP case will be modified. Right now, what you're doing is changing the age, therefore changing the decay of the NMDA because you're having more slowly NMDA when you're young, a little bit faster NMDA when you're old. And here we don't see a sliding of the threshold. The separation is still clear in 10 hertz. We notice when using the same experimental conditions for Dudek and Bear93 that only change magnesium, it is able to make this sliding threshold to occur. So here you have the 1.5 millimolar of magnesium, around 10 hertz. And if you increase it, if you decrease it, you will start to have this drift in the sliding threshold. So I'd like to show a simulation. First, for the 1 hertz at the definition of the experiment, that's 1.5 millimolar of magnesium. And yes, as I showed before, there is no chance for it to reach the LTD region. However, if you decrease just a little bit, just 0.5, you are able to reach it, okay? And then you can produce a very robust LTD. And again, if you decrease even further, you can have a much larger activation of NMDA. That's very much. So right now, I'd like to finalize with